This lesson is about non-coding DNA. Non-coding DNA is DNA that doesn't code for a protein product. And there's lots and lots of non-coding DNA. In fact, by some estimates, the human genome is as much as 98% non-coding DNA and only 2% coding. We can think of non-coding DNA as divided into two groups: DNA that's still transcribed even though it doesn't make a protein product, and DNA that's not transcribed at all. Remember, transcription is taking a DNA template and making RNA from it. So if it's transcribed but it's not made into a protein product, what kind of RNA could it be transcribed into? Well, it could be transcribed into ribosomal RNA, the RNA that makes up ribosomes together with proteins, and it could also be made into transfer RNA, which is used in translation. Let's now take a look at a few different kinds of non-coding DNA that's not transcribed. This is a drawing of a metaphase chromosome, a 2-chromatid chromosome, and I'm going to use that to illustrate a few different kinds of non-coding DNA. First of all, you're already familiar from mitosis with the idea of a centromere. A centromere is a non-coding region of DNA in the center of the chromosome, and that's where the spindle fibers are going to attach in mitosis as well as in meiosis in order to pull the chromosomes or chromatids to opposite poles. We've also seen that throughout the chromosome there are origins of replication. Remember that origins of replication are those places where the DNA is going to separate to form a replication bubble. So here's our DNA as a double strand, and then it opens to form a replication bubble, that bubble consisting of two replication forks that proceed in opposite directions. And there are lots of these origins of replication throughout eukaryote chromosomes. At the end of the chromatid arms, there are also telomeres, which we will talk about in a moment, and they form caps at the end of the chromatids. Another kind of non-coding sequence in eukaryotes are introns. Remember that in eukaryotes, the nucleus separates transcription, that is, making RNA from a DNA template, from translation, using the messenger RNA to make a polypeptide chain. And because of that, we can have another step that goes between transcription and translation, and that's processing of the RNA. So there are long sequences in the DNA that never make it into the mature messenger RNA. What I've drawn here is a precursor to the mature messenger RNA that's never going to make it out of the nucleus, and this is called pre-mRNA. And here, this contains complementary bases to all of the nucleotides that were in the DNA. It's just been transcribed straight off the DNA. But there are sequences here, and these are called introns, that I've drawn in red, that are not going to make it out of the nucleus. They're going to get cut out. And the sequences that make it out of the nucleus, that get cut out, are called exons. So exons are going to make it into the mature messenger RNA. They're going to leave the nucleus, and then they're going to be used at ribosomes to make a polypeptide chain. The introns are going to get cut out. In effect, what you're going to see is these three get spliced together to make a mature messenger RNA. And there are other steps in making the mature messenger RNA that we will talk about in another lesson. But this step is called splicing. So introns are these sequences that get taken out and never make it into the mature messenger RNA that's going to leave the nucleus. And we'll talk about some advantages to that in a future lesson. By the way, I've drawn exons and introns here on the messenger RNA transcript. This is meant to represent a single-stranded molecule of pre-messenger RNA, but intron and exon are also used for the DNA sequence from which this has been copied. So you'll hear intron and exon used to talk about DNA as well. Now let's come back to telomeres. So I said before that telomeres are regions at the ends of eukaryote chromosomes. Let's investigate more what they are and what they do. So this is meant to represent a DNA double-stranded molecule. And let's make this the 5' end of this strand and the 3' end. So this is the 5' end and the 3' end of the complementary strand. If we imagine this going through one cycle of replication and separating and then complementary base pairing happening, if we think about what happens at the ends of the chromosomes, we notice a problem. Now, I've drawn this very much not to scale so that we're going to see both ends of the chromosome here, because that's just what we want to focus on. So keep in mind this is very much not to scale. Remember that because DNA polymerase can only work in the 5' to 3' direction in terms of the polymer that it's laying down, we need a primer for it to start off of. So here in green, I've drawn the primer on each of these strands, and I've drawn the new strand of DNA in a bit of a lighter shade of the same color as the template that it was using. So the primase started here, laid down an RNA primer going in that direction, and then DNA polymerase continued after that. And the opposite happened over here. And so normally what would happen on the lagging strand is that you would have this replaced by DNA, and then you'd have DNA put down and ligase would join the sugar phosphate backbone. But at the ends of the chromosomes, we have a problem, because if you remove this, and that's what's seen happening here, or when that is removed, you can't now go back in and add a DNA replacement for that primer, because there's nothing for it to hang on to. There's no 3' OH for it to grab onto. And so you just end up with this blank region and this uneven ending chromosome. And on the other end, you'd have the same thing. Again, very much not to scale, but you get this missing bit at the end of the chromosome. And now if you were to imagine this separating again and going off and doing another cycle of replication, you'd see that this shortening problem gets worse each time you replicate. And so the chromosome is basically being sort of chewed inwards from the ends with each replication. So what telomeres are, is they are these long sequences of repeated nucleotides, long repeated nucleotide sequences at the ends of the chromosomes. And they actually act as sacrificial DNA, and they are what's being gobbled up at the end of the chromosomes. And by putting that DNA in the way, those repeated sequences that aren't part of genes, they get gobbled up and the genes get protected. So it's the telomeres that get shorter, the telomeres that shorten with each division. Now, if a cell divides a lot, over time those telomeres will get too short, and soon you would start to encroach on the genes. So when telomeres get too short, they trigger the cell to die. They trigger programmed cell death, and so the cell will die rather than continue to replicate with damaged DNA, which could be dangerous and cause cancer. By the way, that kind of programmed cell death is called apoptosis, which is spelled like this. You'll also hear it pronounced apoptosis, and that's the term for programmed cell death. Effectively, the single cell in a multicellular organism is dying rather than jeopardizing the whole organism. Now, germline cells, like those cells that go to make sperm cells, need to replicate a lot, and so they have an enzyme called telomerase, and telomerase actually adds nucleotides to lengthen the telomeres in order to keep the chromosomes at basically the same length, so that as they're being chewed up, they're being replaced. And that allows those cells to go through multiple divisions without triggering programmed cell death. Now, cancer cells also often have high telomerase activity, and you can reason that out because if they didn't, they would tend to kill themselves off, and cancer is effectively a kind of immortal cell. So they've often developed mutations that keep telomerase working. So that's an introduction to non-coding DNA.